afternoon and welcome. My name is Chandrakant Lahariya and I am moderator cum speaker for this webinar. Before I start this webinar, let me start with some disclaimer. First, everything which I would say during this webinar is my personal opinion, should not be attributed to my professional affiliation. And second, I would like to make disclaimer that I'm not being paid to speak here by either Practo or any organization. I have no conflict of interest. So, as per the format of this webinar, as I'm the speaker as well, I'll introduce myself first. I'm a medical doctor by training and I'm a public policy and health system specialist. In the last 15 years, I have worked mainly in the disease control and elimination. My work had been on a polio elimination program as well as routine immunization spending. I worked with the national government, not in the government, but with the national government in introduction of number of new vaccines as well as developing and drafting the national policies. In addition, in the last few years, I have been working on health system strengthening. And for last 1 year, when everybody is working and putting the fight to COVID-19 pandemic, I have been involved in that activity. Just another note that my book on COVID-19 pandemic, titled as Till We Win, India's Fight Against Pandemic, will be released on 23rd November 2020. It's a co-authored book and it's available for pre-order on various platforms. So this is my introduction. I'll be moderating this session and I have two esteemed panelists and speakers whom I'll introduce in the due course. As we are talking in this webinar, today, 31st October is exactly 10 months since the first case of COVID-19, that time known as the novel coronavirus, was reported from Wuhan in China. Today, we have around 45 million cases, more than 1 million deaths, and there is no end. Rather, a few days ago, more number of daily cases were reported from the world than any other day in the past. The COVID-19 is spreading. Europe is facing a second wave, and though fortunately in India we are seeing a decline of the cases, but situation is nowhere under the control. What we also know that there is no effective therapy available, no proven therapy available till now. What is working for us is public health measures and non-pharmacological interventions such as face masks, hand washing, and social distancing. In this backdrop, everybody is asking question, when the pandemic will be over? And no one knows the answer to this question. In this setting, there is one hope, the hope of the vaccine. And all of us want a vaccine as early as possible, and hopefully that will be available soon. So the theme of this webinar is COVID-19 path to an effective vaccine for India. As we talk about this aspect of pandemic, we will talk about three aspects, three dimensions. When we want to talk about vaccine, we need to understand the virus and the immune response it creates. So that's a one stream, and that's the first presentation we will have. And then we would focus upon vaccine development. That's a second set of presentation. And then development of vaccine should lead to the delivery of vaccine. So third part of presentation or third set of presentation would focus on vaccine delivery. I have two eminent speakers and myself as one more speaker for this session. We will distribute each of my co-panelists or speakers will make a 15 minutes short presentation. After three presentations, and we will go for a question and answer session, which will last for another approximately 45 minutes, and we will conclude the session. So without further ado, I would like to invite my first panelist and speaker, Dr. Ravi, who is the senior professor and head of neurovirology at National Institute of Mental Health and Allied Neurosciences in Bangalore. He's also nodal officer in Karnataka Health Task Force for COVID-19. Dr. Ravi is an internationally respected virologist who has more than a few thousand citations in national and international medical journals. His areas of interest include public health virology, development of indigenous kits, antiretroviral therapy, immunology, pathogenesis of viral infections of CNS, central nervous system, and psychoneuroimmunology. Dr. Ravi would be talking about the virology and immune response to SARS-CoV-2. Thanks, Dr. Ravi, for agreeing. Over to you. Your 15 minutes start now.
Thank you, Dr. Lahariya, and good afternoon to everybody. It's a great pleasure and honor to speak in this webinar. I thank Practo for having me here. What I will do in the next 15 minutes is to talk to you about virology, immunology, and immunopathology of COVID-19. I hope you're all able to see the screen. Can you confirm, Dr. Lahariya?
Yes, we can see your screen.
Okay. I have disclaimers. SARS-CoV-2 is a new virus and we do not have sufficient information on the virus fully or its immune response. We therefore have to rely on the limited information available. It's a very dynamic situation. Things keep changing every day. This presentation will use information only from peer-reviewed journals, indexed and available on PubMed, not from predatory journals. I certainly will not use information from social media, especially from the world's largest unregulated university known as WhatsApp. I have no conflicts of interest and I have no commercial interest. And like Dr. Lahariya said, we are not paid for this symposium. What I will do in the next 12 minutes is to talk to you about virus structure, antigenicity, basic biology, a word about the clinical spectrum, introduction to immune response, the basics of immune response, both innate and adaptive responses, a word about immunopathology and vaccine pathways. I'm sure my subsequent speaker will end up with summary. What do we know about coronaviruses? They have been around for 50, 60 years. They are basically viruses that have an envelope. Not all viruses, as you know, have envelopes. This is an envelope virus. Second, it's an RNA virus. Viruses can have either DNA or RNA, and this is an RNA virus. And they are widely distributed in nature, in humans, animals, birds. Prior to the occurrence of this SARS-CoV-2, the novel coronavirus, which came in 2019, we knew 6 other coronaviruses, 4 which have been designated by numbers and alphabets like 229E, OC43, NL63, HKU1. All of them cause mild infections, usually upper respiratory, and some of them cause mild gastrointestinal symptoms as well. The first serious coronavirus came in 2002-2003. It produced an severe acute respiratory syndrome. That's why it was abbreviated and called SARS virus. In 2012, we had the Middle Eastern respiratory syndrome. To distinguish from SARS, it's a different virus. Geographically, it is present in the Middle East and some other countries. Fortunately, both these did not enter India. Since they are widely distributed in nature and they exhibit genetic diversity, quite often what happens, these viruses spill over or cross species from man to animals or birds and vice versa. The name coronavirus came because under the electron microscope, it is spherical and its surface is studded with projections looking like the crown of a king. That's the way the name corona came. Bats and rodents are the natural reservoirs in nature. From these natural reservoirs, periodically spillover occurs to other species, maybe animals, maybe birds, and then from there it spills over to human beings. SARS-CoV-1, the one that occurred in 2002, is now called SARS-CoV-1. That was a spillover to pigs. MERS was a spillover from bats to camels and then human beings. And the latest SARS-CoV-2 is a spillover from bats to pangolin, which is an ant-eater family. The WHO coined the term coronavirus-induced disease 19. To the numbers 19 are given to identify the year in which it was identified. And the disease description is coronavirus-induced disease. This is to avoid any stigma to any nation. The International Taxonomy for Classification of Viruses designated this virus as severe acute respiratory syndrome, coronavirus 2, to distinguish it from the one that occurred earlier this century. The virus itself is spherical. It can measure anywhere upwards from 60 nanometers up to 200 nanometers. You can see it under the electron microscope. It has an outer envelope. In the cartoon on the left, it is depicted as a blue circle. Embedded in the envelope are many proteins. The most important protein is the common pin-shaped protein you're seeing, known as the spike protein. It has a globular head and it has a long stem embedded in the envelope. This is the most important protein. We'll come to it shortly. Apart from this, there is membrane protein. Inside the envelope is the virus shell, which is made up of the nucleocapsid protein, again an important protein, and embedded in the envelope is a very unique protein known as envelope protein. The genetic material is RNA, it is single-stranded, and it is positive polarity. What is positive polarity? When you introduce this RNA into a cell, it can directly go to translation and make proteins. Negative stranded viruses are when you introduce RNA into a cell, they first make a copy, much like the older photo negative where you take a print to make the photograph. Those are negative, but this is a positive stranded virus. On the right is a cartoon depicting the genome of SARS-CoV-2. It is about 29,000 nucleotides in length. It has several genes. The prominent ones are open reading frame 1A and 1B, then the spike protein gene, then you have the matrix and the envelope and the nucleoprotein and so on. And then of course, a gene coding for the RNA polymerase. So this is the genomic organization. How do coronaviruses enter cells? Like all viruses, enter cells through a specific molecule on the cell surface, which we call receptor. And in the virus, there is always one protein or a couple of proteins that are responsible for unlocking the cells. Virus receptor interactions are very much like lock and key. The key coronavirus SARS-CoV-2 uses is the spike protein. It locks into a physiological molecule known as angiotensin converting enzyme 2. This is a molecule present in cells for binding to angiotensin. You all know angiotensin is a hormone that regulates human blood pressure. So in nature, this is a receptor meant for angiotensin, but coronavirus 2, SARS-CoV-2 uses this protein. It then attaches, merges with the cell membrane, and it is internalized into the virus. So that is this spike protein is the key, as I mentioned, and it's a very important protein. The full three-dimensional structure of spike protein is shown on the left. It is a globular head and it has a stem. In the globular head, there is an enlarged portion showing the region where it binds to the receptor, abbreviated as RBD. That means receptor binding domain. Now, this is a very important protein, and this is also responsible for the main immune response that you see to this virus. So antigenically, it is very important. In terms of biology, it's a very important protein to enter into cells. So this is quite naturally a very, very common target for vaccine production. When viruses multiply in cells, they make a lot of proteins which ultimately don't go into the assembly. Now, these are called non-structural proteins. A table on the right shows you NSP is an abbreviation for non-structural protein. As many as 16 non-structural proteins are there. These are found only inside infected cells. They are not found in the viral particle. They have specific roles for either processing proteins or making proteins or copying nucleic acid of various functions. But very interestingly, the ones circled in red are 3 non-structural proteins. Actually, there are totally 5. They have a very profound effect on human defense system or the immune response. They promote cytokine expression, they promote inflammation, they evade normal immune mechanisms, be it innate or be it adaptive. So these proteins are very unique to SARS-CoV-2. If you look at how does coronavirus cause disease, basically it is the disease that you see in coronavirus is not due to virus killing cells. That of course is there, it is very minimal. But the main reason for people developing moderate or severe disease is their host response. Their defense system responds abnormally, producing a lot of cytokines or inflammatory proteins, which then flood the lung. And these cells infect endothelium, so they cause damage. There it becomes a nidus for small clot formation, and thrombi are formed, and that is another very important effect coronavirus produces. Like Louis Pasteur said on his deathbed, the germ is nothing, the terrain is everything, meaning in this context that the immune response is the one that causes infection. The coronavirus disease ranges from asymptomatic to severe. 80 to 90% develop mild or asymptomatic infection, 10 to 15% severe, and 5% critical. And on the right, you see a cartoon which tells you that in the last 10 months, we have understood a lot about this disease because ACE2 receptors are found all over the body in many organs. The virus can infect all of them, and you will get neurological, renal, hepatic, gastrointestinal, name it. The current adage is if you know coronavirus, you know clinical medicine. Moving on to the basics of immune response. As you all know, there are two types of immune responses God has created in the human body. One is called innate immunity, which is the first level defense. They are the soldiers at the borders who attack enemies. And then is the adaptive immune response. What is innate immune response? It is non-specific, meaning it does not distinguish virus, bacteria, fungus. Any invader it will attack. It is present at all times. It is immediate, but general in nature, not specific to a virus. And it has linkages with the adaptive immune response. The adaptive immune response is not immediate. It comes in later. It is more specific. It takes clues from the innate system. The response is very specific. That means the immune response to one virus is not the same, and it cannot be effective against another virus. And it is mediated predominantly by lymphocytes. They are classified as T cells and B cells. T cells, broadly speaking, promote cell-mediated immunity. B cells broadly produce antibodies. And the communication molecules within the immune system are proteins that are called cytokines and chemokines. They are the ones that give signals. So when SARS-CoV-2 enters, it initially multiplies in the nose and throat, then goes down to the lung. It infects type 2 pneumocytes. And if the innate immune response is able to eliminate the virus, it's fine. Otherwise, it gives signals to adaptive immune response. Then come the T cells, B cells, and the inflammatory markers. And then you have flooding in the lung. And that's how you get pneumonia. B cell responses have been detected in SARS-CoV patients within 4 to 8 days. The first response is to nucleoprotein, but what is important is response to spike protein, because antibodies to spike protein neutralize the virus and then prevent it from multiplying and spreading. So neutralizing antibodies are usually directed against the spike protein. Antibodies can also be rarely or occasionally harmful, especially if they're antibodies produced to old coronavirus infections. They will coat the virus, and because it's coated, it will get opsonized or swallowed by macrophages easily, because macrophages have receptors for antibodies, and then it produces what is known as antibody dependent enhancement. This is more with cross-reactive responses. Does everybody produce antibodies? The answer is no. 80 to 90% produce antibodies. It takes 14 days for antibodies to be detected. For some reason, coronavirus immune response in terms of antibodies is very slow. We do not know. Many other RNA viral infections, within a week, you get an antibody response. Here is a paper published in a very reputed journal from Mount Sinai Hospital, which showed that 81% developed antibodies in 14 days. The remaining were followed up for up to 6 to 8 weeks. Even in those who did not develop response, again, only 80% developed antibodies at the end of 8 weeks. So there are some people who do not develop antibodies. T-cell responses have been well characterized in COVID-19. Both CD4 and CD8 cells are elevated. Regulatory cells are less. That's why the immune system goes on a rampage. And we are now realizing that T-cell responses are also very important. It's not only antibodies, but T-cell responses are also very important. Innate responses are the main key. And there are beautiful studies where people have collected bronchoalveolar lavage fluid and looked at the kind of genes that are upregulated, and they found that inflammatory genes are upregulated in the lung innate cells early in infection and in people who have severe infection compared to other viral infections. So this basically is a cartoon that shows you how virus fluid accumulates in the lung, depending on whether the disease is moderate or severe. And what happens is, if in the initial innate or initial adaptive response is able to eliminate virus through antibodies or through cytotoxic cells, then the amount of fluid collected is less. If more cells come in, more cytokines are produced, the whole lung becomes a bag of inflammatory fluid which can enter the blood and then spread and cause systemic manifestations. Traditionally, vaccines take anywhere between 3 to 6 years. The COVID vaccine pathway is very accelerated. I'm sure the Raches, the next speaker, will be able to throw more light on you. We are compressing the entire thing into 12 to 18 months. I hope we will be successful, and there are very promising candidates down the line there. There are various stages which Raches will call about. There are 174 vaccine candidates listed in the website, and there are many that have entered clinical trials. The Oxford vaccine, the Bharat Biotech vaccine, which Raches will tell you about, and there are many other vaccines which are entering phase 3 trials. We'll have results by the end of the year. I hope I've finished in time. Thank you very much. All the others we can take up in the questions. Over.
Thank you very much, Dr. Ravi. This was really comprehensive, and you covered a broad range of disease epidemiology and virus characteristics. But most importantly, you laid the foundation for the next talk by providing the background information on how immunity develops against the disease. We will take questions from audience a little later. And so without further ado, I would like to invite Dr. Raches Ella. He is the project lead for SARS-CoV-2 vaccine at Bharat Biotech International Limited. This vaccine, Covaxin, is being developed by Bharat Biotech in collaboration with Indian Council of Medical Research and National Institute of Virology. Dr. Raches leads this work, and of course, there is no better person than him to talk about this vaccine and all other vaccines which are being developed against SARS-CoV-2. Just for the audience interest, Dr. Raches is a postdoctoral fellow from the Johns Hopkins Bloomberg School of Public Health. The topic of his talk is COVID-19 vaccines, the trials and tribulation. Over to you, Dr. Raches.
Thank you, Dr. Lahariya, for that introduction. My name is Raches Ella, and I'm the project lead for Bharat Biotech SARS-CoV-2 vaccines. Simultaneously, I also head business development and advocacy at Bharat Biotech. Before starting off, I would like to thank the entire Practo team for inviting me here and allowing me to share our results and a few of the concerns and tribulations we have with regards to conducting clinical trials, especially for COVID-19 disease. My topic for today is going to be summarized into three subheadings. The first is going to give you a short snapshot as to what are the clinical trials that are ongoing and what are underway. And then with regards to operationally conducting these clinical trials, what are the considerations we take as vaccine manufacturers, and what are some of the risks that we foresee and how we're trying to mitigate them. And also, I'll be touching base on our co-vaccine development. So to date, this is a New York Times article, which is an update day to day. This gives you updates as to what's going on with the COVID-19 vaccine landscape. I find it extremely helpful. As Dr. Ravi mentioned, there are over 150. I would say, I think the latest number is 190 vaccines from preclinical to phase 2 clinical development that are in development. And we have about 11 or so in phase 3 clinical trials right now. Numerous vaccines are in phase 3 clinical trials. These are efficacy-based clinical trials, and I'm sure you've read a lot about them in the media. And the most recent update was coming from India, where Bharat Biotech had announced that its lead vaccine has just entered phase 3 as well. So there is hope at the end of the tunnel. We just need to be patient, and we just need to ensure that all these trials are done with utmost scientific integrity. Coming to the second part of my talk, which is the tribulations aspect of running these kinds of clinical trials. This infographic here is quite self-explanatory. It's trying to let you know exactly what we go through as vaccine product developers. We first start off with an exploratory R&D stage, which takes anywhere between 6 months to a year. In fact, a year is not the upper limit. Sometimes, it takes time to get the molecule right. So it takes time up till 5 years or so. And then preclinical trials, and then you enter the human stage, which lasts anywhere between 1 to 7 years, I would say. And even after you get your regulatory approval, the buck doesn't stop there. You need to constantly manufacture the vaccine at high quality, and you also need to uphold the safety profile of the vaccine. So you need to do continuous phase 4 post-marketing surveillance studies to ensure that when this vaccine is being rolled out to the masses, and we're talking about billions of doses, there is no untoward effects to the patient. And to summarize here, I would just like to bring one statistic, which not many people are aware of, is the probability of success for developing a vaccine. There are very few vaccine manufacturing companies in the world and in India. There's only one reason for that. The risk is too high. There's about a 6% chance from the exploratory phase. If you find a candidate, there's a 6% likelihood that you will see it licensed, and then you'll be able to sell the vaccine. So there's a huge amount of risk involved, and I'm just trying to lay these expectations out to my physician community here to know that every vaccine you see in the media, there is a chance of failure as well, be it any vaccine. So another concern I had, I'm just, I tend to read a lot of PubMed and literatures, but of course, sometimes the headlines grab me. And a few of the headlines I felt tend to be drafted out of line. For example, you would have heard of AstraZeneca's clinical trial in Brazil, where they mentioned that a participant in the clinical trial dies. They don't give the cause of the disease. They don't give any details, but the headline is very eye-catchy. It's worthy enough for you to click that button and read that article and probably see 2-3 of their ads. But I feel the media is getting this wrong, and I feel this is where the physician community should step up, analyze the data, and try to at least understand the details of the article and try to synthesize this in a much more better manner. And I'm at least glad that the Washington Post carried the right headline, where they mentioned that this volunteer who died in this clinical trial did not receive the vaccine, but they received the placebo. So they didn't receive the AstraZeneca vaccine. They probably received a placebo, which was just sterile PBS. So I think it's very important for the media, and again, for the physician communities, physicians out there, to get this information out so that people understand a little bit better. Another troubling statistic I came across in a European journal, which was a recent survey from a lot of healthcare workers. This contained around 400 doctors, 211 nurses, and 1,000 from the general population. And they asked a few basic questions. The first question was, do you vaccinate against seasonal influenza? And the doctors here answered very well. 92% of them said, yes, they'll take a flu shot every year. The troubling question is when the next question was, would you take a future COVID-19 vaccine for yourself? And that number dropped to about 78%. In fact, and the general population, 75% said, yes, they'll take the vaccine. And another even more troubling statistic is, would you take any COVID-19 vaccine for your child? Would you give it? Would you administer it? And only 60% of the doctors said, yes, they'll do that. So I feel there is a lot of skepticism, no doubt, definitely clinical development is being rushed for these COVID-19 vaccines. But I feel that the manufacturers out there are doing it for the right reasons. And as mentioned before, we're going to uphold scientific integrity, and all data should be published in publicly available websites for you to see and be comfortable with at least the safety and the efficacy results. Another interesting statistics coming to the same paper was the discrepancy between the internal medicine physicians and the general surgery physicians. 91% of the internal medicine physicians said, yes, they'll take the future COVID-19 vaccine, whereas 75% of the general surgeons or the surgeons, super specialist surgeons, said, no, they won't take the vaccine. So this depends on what environment the physician is in, probably, and definitely how comfortable they are with the vaccine. But I just thought these are some useful statistics for us to curb vaccine hesitancy that will come for COVID-19. And I think it's a very important issue, and it should be tackled with a great amount of detail and care. I don't want to stress too much, as Professor Ravi alluded to in detail, about another concern we have is about this phenomenon called antibody-dependent enhancement. It's basically when you get this, in theory, it works as you get the COVID-19 vaccine, you have some amount of antibodies, but over time, those antibodies decline. And when they decline, you're more prone to getting COVID-19 disease, and that exacerbation is just of another level itself. It's more serious. So I think this is also a huge concern. There are reports of an earlier SARS-CoV-1 to have this kind of phenomenon, but this was in preclinical models. This was like in cats and in chimps. But we're working aggressively to understand the immune responses of our vaccine and to make sure that this does not repeat again. And again, I don't want to repeat myself, as Professor Ravi had alluded to previously, but I'm just trying to summarize, in essence, two things this COVID-19 vaccine needs to do. Number one is it needs to produce antibodies. These antibodies are of neutralizing in nature. Basically, that means they block infection, which will offer some amount of short-term protection. The vaccine immune response will wane over time. As you can see, even in naturally infected COVID-19 patients, their immune response plummets after 3 to 4 months, I would say. So it's very important for any vaccine to induce a T-cell response. This T-cell response will, in theory, at least protect these people for much longer durations of time. Coming to Covaxin, and a little intro on Bharat Biotech. We're quite a young vaccine manufacturing company. We're 25 years old. We have around 16 different licensed vaccines, ranging from the pediatric cohort to the geriatric. We take pride in doing our own clinical development. We've done around 80 clinical trials across 18 countries, including many countries in the African Union, the UK, and the USA as well, in over 600,000 volunteers and 500,000 infants, newborn infants. There's a lot of talk going around about human challenge study. In fact, we were able to partner with the Oxford University to do a typhoid conjugate human challenge study about 5 years ago. And it was based on the results of this. Our vaccine was licensed, which is the Tyvar TCV. It was licensed in a very short duration of time. So there are some pros and cons to human challenge studies, but for the purpose of this discussion, I don't want to get into it. And over the last 25 years, we've sold around 4 billion doses across 120 countries. And our goal is to only increase that. And with COVID-19 in mind, we are quite confident of doing that. And also, as mentioned to you before, there's a lot of risk when you try to develop a vaccine. And this is not done only by Bharat Biotech. I mean, it's a team effort. We consider this, and we truly value our partners who have been with us along the way to develop these various vaccines. Coming to COVID-19, we have around three different strategies for COVID-19. The first is in partnership with the ICMR India, and the second is with two US universities, the University of Jefferson University and the Wisconsin University. And we're trying to develop three different COVID-19 vaccines. With regards to Covaxin, it's a whole virion inactivated vaccine. So I'm sure you would have heard about various terminology, mRNA, DNA, adenovirus vector-based vaccines. These are all quite new vaccines and they've not been licensed before based on these platforms. So we try to keep things simple. We tried to use this approach called whole virion vaccine, which basically means you take the virus in these large bioreactors, these fermenters, you grow them to an adequate volume or a viral concentration, and then you inactivate them with a chemical agent. This is a very trial and tested method. It's been there for the last 5 decades or so. It's conventionally very safe, extremely safe, I would say. It can be used in special populations such as pregnant individuals, elderly, or even immunosuppressant individuals as well. And there's various vaccines that are already licensed based on this platform: polio, pertussis, seasonal flu, rabies. So we thought this would be a no-brainer for us to work on this platform to quickly develop a COVID-19 vaccine. And also another bolstering parameter for us was we've done this before with several of our licensed vaccines. We have a rotavirus, Japanese encephalitis, and rabies vaccine, which is already licensed. And we also have a few vaccines that are in clinical development, such as the chikungunya, Zika, and an injectable polio vaccine. And overall, we sold 300 million doses of these vaccines based on this Viro cell manufacturing platform. So we thought we could adapt Covaxin based on this platform, and hopefully it would generate some good results. Another quite unique thing about Bharat Biotech is we have a special facility which was made for another vaccine, but it's been repurposed for COVID-19. This is called a BSL-3. BSL stands for Biosafety Level 3. It's a production facility, not a research lab. So this is the only BSL-3 production facility in India, and I think there are around only 5 in the world. So it's for those reasons alone, my colleagues, as you can see in this picture, are able to handle the virus, even hold a Petri plate of the virus in their hand. As you can see, they're completely donned with their PPE, and this facility has been audited before, and it's currently manufacturing Covaxin as we speak. Coming to Covaxin's progress, it's branched out into 2 parts: the preclinical, which is the animal studies, which was done in rabbits, mouse, and rats, and also monkeys and hamsters. And we've also done 2 phases of clinical development, and we just received our 3rd phase of clinical development to begin, which will begin very shortly. With the preclinical studies we've extensively done toxicology and immunogenicity studies. And as you can see by these graphs here, that the vaccine was able to create an antibody response and also a T-cell response, which is very important, as Professor Ravi highlighted. These results are published on bioRxives, and I encourage you to take a look at them. Coming to live viral challenge study. So this is quite unique, especially in India. I don't think it's been done before with a vaccine. We were able to partner with the ICMR, National Institute of Virology, Pune, to conduct these studies, where we are the first vaccine to do 2 of these studies separately. But for the pros and cons of these animal models, we decided to do it in 2. So we did it in monkeys first, and then we also did it in hamsters. And these results are available online as well, the preprints, and the full publications also will be published very shortly. The whole concept works like this. You take a few animals, like monkeys, for example, in this instance, non-human primates. You give them 2 doses of the vaccine. After 2 to 3 weeks, you in fact challenge them intranasally and intratracheally with live COVID-19. And then you monitor them over time to look at the viral clearance, which is RT-PCRs, no swabs, and then you RT-PCR them. And you also look at disease. Does the animal succumb to COVID-19 related symptoms? So it's a very good way of getting a short snapshot as to how the vaccine is working and is the immune response protecting. And as you can see by these graphs over here, post-challenge, so that means once the animal was vaccinated and then they were challenged with COVID-19, days post-challenge, after these challenge, on days 3, 5, and 7, you're able to see that the amount of virus circulating in the upper airway and the lower airway falls dramatically in the vaccinated group. And in the placebo group, it's still present. So this is a clear indication that the vaccine is helping fight the virus in the upper airway tracts and the lower airway tracts, which is very important. We've done a very similar approach in hamsters as well. And for the interest of time, I'll skip this, but in summary, the vaccine is doing two things. It's protecting in the upper airway and it's protecting in the lower airway. Two very important aspects for a good COVID-19 vaccine to be efficient. And coming to clinical trials and vaccine efficacy, if you ask me, this is a personal weightage I've added here. And coming back to these two little fundamental concepts, the vaccine needs to prevent disease, of course, but it also needs to prevent infection, and thereby it will prevent transmission. So I think these are things which we have to incorporate as product developers into our clinical trials. Covaxin's gone through a phase 1 clinical trial, intramuscular approach, and we've also done an exploratory pilot stage intradermal approach as well. There are many benefits for doing an intradermal approach, and we've also successfully completed that trial. We've completed vaccination of our phase 2 clinical trial, and we're generating the interim results as we speak. And last week, we've received permission to start our phase 3 efficacy-based clinical trial. Coming to the phase 1 clinical trial, we're trying to evaluate 3 such formulations of Covaxin with a placebo arm as well. This was done in 18 to 55-year-olds. They all received 2 doses of the vaccine. This was a phase 1 double-blind multi-center clinical trial to evaluate the safety first. The secondary outcomes were immunogenicity, but the primary outcome was only safety, just to make sure the vaccine was safe. We've conducted this clinical trial in over 11 different hospitals across 9 states. We're trying to make our results as generalizable as possible to the country. There's a lot of diversity and differences across state to state. So we wanted to prevent that creeping into our analysis. Coming to the phase 2 clinical trial, we were finally able to select 2 such formulations, the BBV152A and the B formulation. We expanded our age cohorts to include a certain section of the pediatric cohort and a certain section of the geriatric cohort. So it's 12 to 65 years. This is also done in 380 participants, where here we're primarily looking at the immune response, and the secondary outcomes are also evaluating the safety of the vaccine. Most recently, we received our approval by our DCGI to start our phase 3 clinical trials. This will be done in 26,000 volunteers. I made this slide just to give our viewers a slight idea as to what these trials are. So, there's a lot of vaccines can call themselves a phase 3 clinical trial, but we're doing something called a phase 3 efficacy trial. So what that means is we take 26,000 participants, enroll them into our study. We randomize them equally to receive either Covaxin or they receive a placebo. And we're all blinded to this. The principal investigator, the healthcare workers, and even us, we're all blinded. We don't know who gets what. And over time, we monitor them up. We call them every 15 days and we inquire if they've succumbed to COVID-19 disease. And we aggressively try to collect samples from them periodically to see if they have been infected with COVID-19. And at the end of the study, we compare what is the incidence of disease in the vaccine group, what is compared to the incidence of disease in the placebo group. This is how we gauge vaccine efficacy. And this is why we need to do it in such a large sample size of 26,000 to be specific. And ultimately, we're trying to, if any vaccine manufacturer would love to see a graph like this, where once we follow up these participants in the placebo arm, in theory, if they have double, say there are 100 COVID-19 diseases in the placebo arm, and there's 50 COVID-19 diseases in the vaccinated arm, that would go to show that the vaccine is efficacious by at least 60 and beyond %. So that's something very important for us. But also another concern we have is getting these critical people. So I think the most important purpose of a vaccine is to save lives, most important. So I feel any vaccine should be targeting this group, the critical and the severe group. So it's very important that we're able to gauge the efficacy of these vaccines for the critical and severe people as well. So that's another reason why our sample sizes are quite large, 26,000 to be precise. To summarize, there are numerous challenges for conducting clinical trials in India and the world as well. Low disease incidence is a very troubling statistic. You can't just do a phase 3 trial anywhere in the world. You can't go to, say, Sri Lanka, which have very low disease, or probably Thailand. You need to do it where there is ongoing circulation. So a place like India is quite ideal to do this. Antibody dependent enhancement, vaccine, or it's also known as vaccine associated enhanced disease, is a concern for all of us, and we are evaluating it extensively. There's also the fundamental criteria that this vaccine needs to prevent against disease, be it severe or mild, and also infection. And also, from an operational standpoint, vaccinating 26,000 volunteers, following them up to ensure that the vaccine is safe, there is no untoward side effects, and the vaccine is working is a huge challenge as well. Bharat Biotech is the first company to conduct such an efficacy trial for COVID-19 disease. This is the first efficacy-based clinical trial in India, and this data will be very generalizable to India, and we'll be able to extrapolate our results once we roll out these vaccines as well. We're targeting a Q1, Q2 of next year, hopefully, to generate efficacy results. If they are promising, the vaccine could get licensed. If they're not, we'll probably have to extend the study, analyze it further, and make a decision by then. But it could happen, it could not happen. Science dictates these things. It's not vaccine manufacturers. And again, lastly, my point is the onus is on all of us, all of the physician community, I'm stressing to, and various healthcare workers. It's our responsibility to provide clear and uniform messaging to our respective communities so that there's no misinterpretation, especially as Professor Ravi was mentioning about WhatsApp University. I think we need to work hard and work together to dispel all those claims and nonsensical forwards. With that, I would like to thank you, and I'll be happy to take any questions after Dr. Lahariya's presentation. Thanks a lot.
Thank you very much, Dr. Raches. A really comprehensive presentation where you provided information on the landscape of various vaccines under development. You also answered some of the questions indirectly that why we need so many vaccines, simply by laying out on the table that the success rate in the vaccine development is really low, so we need more vaccines. And then you discuss about the vaccines which you are working upon. So thanks again, and there are many questions which I see on the screen, but we will take those questions after I make my own interventions. Since we are already running a little late, so I'll keep my talk relatively short, but we have a lot of questions and a lot of many interesting points to discuss, and we'll come to you. So we started with virus and how it helps in developing immunity in an individual. Then we move to the vaccine development, but development of vaccine is not enough in itself. We need to be able to deliver the vaccine to the people who need it most. So my talk will focus on what should be done and how to ensure that vaccine reaches to all those who need the vaccine. In case of India, it's a 1.38 billion population. How to ensure that we can deliver the vaccine to all these people. Before we move to the delivery part, I want to reflect and build upon what Dr. Raches Ella and Dr. Ravi discussed. That many people keep talking, and many of the people ask me, and many other people are often asked this question: will a COVID-19 vaccine ever be developed? And often the argument made that it's more than 4 decades that there is no HIV/AIDS vaccine. So what is the assurance that we can have a COVID-19 vaccine? What is commonly not understood that there is a difference in HIV/AIDS and COVID-19, because HIV virus as such affects the immune system itself, which is required to develop vaccine or develop immunity. But there is no such thing with COVID-19 or SARS-CoV-2. So we are fairly reasonably confident that there would be vaccine and there would be vaccines very soon. The second issue which is often raised, and people say that in the process of this one of the fastest race to develop COVID-19 vaccine. Are safety issues being compromised? How come a vaccine can be developed in such a short period of time? There is answer to that. So first of all, it is true that not many vaccine or no vaccine has been developed and which is currently used in a short period of time. Often argued that mumps vaccine was developed in a shortest period of 4 years from 1963 when the work started to 1967 it became available. And then probably Zika virus vaccine was at the stage where it could have been developed in 3.5 years. But now with COVID-19, this is not going to happen. However, the fastest race or the approach being followed should not be a cause of concern and worry. There are approaches which are being followed to expedite the process, and of course, we keep hearing that safety is being given as much importance as required. The data safety monitoring boards keep looking at the any safety data. The trials were paused and then restarted, and so all the due diligences are being followed. What is being done mostly to shorten the process that the approving authorities are giving expedited approval, and then sometimes the approvals are being given for combined phase 1 and phase 2 trials. So the time is being saved in those processes, and that's why the fastest race is happening. But more importantly, there are some of the vaccines which are relatively ahead in the race to uh, develop or advanced stage of vaccine development. They are they could achieve that status because some work in the past was already ongoing on those platforms. For example, as we already know that there are a number of vaccines which are uh, being developed on a completely new platform on which no vaccine exists from the past. The Moderna vaccine mRNA platform, by that platform, no vaccine has been developed. However, the advantage here was that on 11th January 2020, the authorities in China had placed the entire viral genome in the public domain. And if you have an entire genome, then the researcher and vaccine experts sat together and they identified that part of COVID-19 or SARS-CoV-2 virus which can generate immune response in an individual, and that part of virus has been taken and will be injected in a individual as part of the vaccine. And it is expected that once that viral genome is injected in human being, the human body will react and develop antibody, and that will work as a vaccine. So when the natural infection happens, it will prevent or protect the infection. So it was the case of viral genome being available, and that's how this is faster. Similarly, the Oxford AstraZeneca vaccine, which is a viral vector vaccine, it could move ahead in the race ahead from other vaccines because the Oxford University was already working on MERS vaccine platform, MERS vaccine on the same platform. So till December 2019, Middle East respiratory syncytial virus or respiratory virus syndrome virus, the vaccine was being developed. But when COVID-19 happened, they quickly moved that platform to develop COVID-19 vaccine. So the technology was already explored and then it was tried for a new vaccine. The another vaccine which received a early license, Sinovac in China, that vaccine is inactivated vaccine. And some of the background work on that kind of vaccine and platform was started on SARS in 2002 and 3, and the researchers had already done some work that how to attenuate or kill the virus in a different settings. However, that work need to be stopped because the SARS, severe acute respiratory syndrome, disappeared. And when there is no disease or no such things, you cannot test such platforms. However, when COVID-19 happened, and this is the same family of virus, and so they could use that platform. So there had been approaches and technologies which contributed to expedite the process. However, we believe that all due diligences are being followed. So we can hope that there would be a few vaccines available against COVID-19. That brings to the question that when will those vaccines be available globally? When that vaccine will be available in India? And the third layer of that question, Anybody can ask, if I'm a healthy adult living in India, when will I get that vaccine? So availability of vaccine globally, within India, and then for a particular target group. So these are the questions which are being asked. They cannot be answered definitively, but we definitely have some clue. What we know that there are a number of vaccines, around 9, 10, which are at the phase 3 clinical trial, human clinical trials, and the data from those vaccines would be available in weeks and months ahead. The success rate is not 100%, but definitely phase 3 clinical trial, which are going on for 10 vaccines or 9 to 10 vaccines, we can hope that there would be a few vaccines, cannot say how many, but there would definitely be a few vaccines which would be found safe and efficacious. And those would be available. So we can expect a vaccine in some part of the world, wherever those vaccines which are found safe and effective are developed, by end of this year or early next year. When can we expect the vaccine in India? Of course, there are indigenous vaccines which are being developed in the country at various phases of clinical trial. There is one vaccine which is being tested in phase 3 clinical trial, which is not indigenous vaccine, but definitely being clinical trial, phase 3 clinical trial, bridging studies uh, sort of are being done for that vaccine in India. There are a few other vaccines which are developed outside India, but are in phase 2 and 3 clinical trial. So what we can say that if those vaccines are found safe and efficacious, we would have a vaccine available for India also almost similar time or with a short gap as global vaccines. But it depends upon which are the vaccine and which country those are developed and found safe and efficacious. And what is the status of a clinical trial of those vaccines in India? If those are being already studied within country, the availability will be likely. But having said all these things, what we can say that there would be some vaccines, and that's a hope, whether indigenous vaccine or vaccine in collaboration with international partners, would be available in India by late quarter 1 2021 or quarter 2. But that's not the end of it. What is required that where the vaccines are developed and the vaccine manufacturers who have done various agreements with the different countries, that will also influence the availability of vaccine. So how many doses would be available would also be determining that how many people will get the vaccine. What we know that most of the vaccines at advanced stage of development would be required to be administered in 2 doses. So definitely the quantity of vaccine needed would be far higher than number of people vaccinated. The quantity, if 2 doses are required, will be exactly twice the number of people can be vaccinated. And that's what we heard a few days ago or a few weeks ago. Uh, Union Minister had said that around 400 to 500 million doses are expected to be available by July 21, which will be enough for 200 to 250 million or 20 to 25 crore people in India. So there are discussions and all these things, plannings or which are being done. Now, the, of course, the question and one thing is clear that once, no matter when the vaccines becomes available, no matter which vaccine available, we need more than one vaccines to have sufficient supply. And if we have an indigenous vaccine, that would have advantage that there is likely to be more availability of vaccines for the country. The cost is likely to be lower for the country, and so that will ensure affordability, availability. But of course, the other important point is that an indigenously developed vaccine or a vaccine developed in India likely to drive the global cost of the vaccines and global landscape, because the vaccine developed in India are safe and effective and they are supplied to rest of the world for other vaccines, and it's going to be the similar setting. So we should really hope that a few vaccines are indigenously developed, which will ensure availability, accessibility. Now, who understanding the point that for a population of 1.38 billion, there cannot be enough vaccine in one go. It can be there in over a period of time, but not in the year 2021. It will be a fixed or finite quantity of vaccine which will be available. Now that number could be anything, but it will not be for all people. So to effectively deliver the vaccine which will become available, which could be three vaccines or five vaccines, pooled number of vaccines, but whatever number is available need to be prioritized for which are the target population which should be vaccinated. So there are some global principles which are laid down by international organizations such as World Health Organization, and also within India, there is a expert group, a national expert group on vaccine delivery, which has already started planning and deliberating these aspects. This is situated within Niti Aayog and Ministry of Health. So those kind of deliberations are happening. What we know broadly, and this information is there in public domain as well, that initial target or initially the vaccine will be administered to the healthcare workers and essential service workers. Now that definition will further be refined. What is globally known and what is broadly known, that the total number of proportion of population which constitute or total number of health workers constitute around 2% of the total population of the country. So the health workers across the country who are involved in COVID-19 fight, which should mean doctors, nurses, sanitation workers, to ward boy, to the uh, the community health worker working the rural area, and those urban settings going for the contact tracing. But not only health workers. So the discourse say that this should be for all the essential services worker, which means people who are in the police services, bus conductors, shopkeepers, all those who are ensuring that country should keep running. Rather, there is a very good editorial in the famous journal Lancet, which said that one of the learning from this pandemic, COVID-19 pandemic, is that the world is run by essential services workers. If they are affected, the world halts or stops. So clearly there is a broader consensus that initial amount of vaccine, whatever amount is available, should be targeted at healthcare workers and essential services workers. Who would be included, excluded, of course, will be decided by an approach and mechanism, but 3% of the population of this worker will be vaccinated first. And the next lot of people who will be vaccinated would be the people with comorbidities. Now, these are the broader principles, and of course, the final call will be taken as and when vaccine will be available, and it is taken through a process. So what we know that the people with comorbidity and high risk people, other than health and essential worker, would be population which is more than 65 years, and anyone has comorbidity like diabetes, hypertension, under treatment. And that would take the number of doses required, which will be approximately 20% of the total population of India. So that's what makes 20%, and that's what being targeted for year 2021. That's what we are hearing. Now, one key principle which we need to remember, that how the prioritization is done or should be done depends upon what we want to achieve. So, for example, the approach which I suggested is mainly to reduce the mortality. If the immediate focus or the focus in particular setting is to reduce the mortality, then the people with the high risk are need to be vaccinated. However, imagine a scenario that there is a second wave, or there is a high rate of transmission in the community. Then one can argue that purpose should not be to reduce the mortality, but to halt the transmission. And if the purpose is to halt the transmission of the virus or pandemic, then the prioritization had to be done for those population who are going out and who are engaged in various services. So to halt the transmission, the approach had to be vaccinating adult population who is going outside the homes and working in different settings. So a lot would depend upon the stage of pandemic and what is happening at that point of time when vaccine are available, and that would determine the prioritization. It is possible that it could be a combination of both. You want to reduce mortality in high risk population, but you want to stop the transmission in those settings. So it is likely that the vaccination approach and strategy vary from one state and one city to another city with some broader principles. Uh, I would wrap up and then we will take some of the questions. The key is that in all countries across the world and also in India, that kind of planning is already being done to deliver vaccine. What is happening? The discourses are happening to increase the production capacity of vials, because if vaccine is available, to fill those vaccines you need some glass vials and there is a finite production capacity. So the discourse is on increasing, assuring availability of the syringe vials to fill the vaccines, the syringes, the training of the healthcare workers, and also to ensure the cold chain. We know that the vaccine need to be stored at a particular temperature. There is one particular vaccine which is a minus 80 degrees Celsius, and probably that kind of setting is not available in India. But most of the vaccine need to be stored at 2 to 8 degrees Celsius, and they need to be stored at that temperature, and that chain need to be maintained always. Rather, one of the key difference in vaccines and cold drinks is that while cold drinks can be stored at the normal temperature and then put in the refrigerator to cool them down, but that is not case for vaccine. Vaccines throughout the chain need to be stored at that particular temperature. So one of the major challenge would be to having sufficient cold chain capacity in the country. And although discussion are happening, and we can, and that requires some planning, and that planning is happening. So finally, to conclude, and then we will move to the question and answer session. Many people are saying that this is going to be a really big challenge and exercise of unprecedented magnitude, which has not been done. And can India do it? Well, the answer is that it may be unprecedented magnitude, but it is not likely to be, because in India there have been campaigns for a measles campaign in 2010 and 11, which had vaccinated around 240 million children, population, 240 million or 24 crore children across the country. Then there have been other campaigns, measles and rubella vaccine campaigns. So country has experience of conducting such mass scale vaccination. Of course, every vaccination is very different from all other vaccination. Those vaccination for measles and MR, measles rubella vaccine, were mainly targeted for 1 year to 15 years of age. There are mass campaigns done for Japanese encephalitis vaccine in the past. Uh, this one, COVID-19 would have a different target population. However, considering the steps involved and the planning involved, country definitely has some experience. So it will be uh, it is not something which we don't have experience in the India, in the country, and it is very much possible. So with these thoughts, I would stop, and I think we are at a stage where vaccine would be a reality. There is some planning going on, and it would also be a reality that soon vaccination would start in the country, and your turn or turn of anyone will come in the process. When, we don't know, but key is that vaccines will help us in moving out or stopping the pandemic in India and across the world. Thank you very much. So I changed my role now, and now I'm going back to my moderator's role, and I hope my co-panelists are here. We have plenty of questions coming on this different platforms. There are most of the questions are related to the vaccine development and other aspects. But I would take a question on start with the order in which we made the presentation. So I would like to pose a first question, probably Dr. Ravi can answer. And they say that there is one question which say that, is this vaccine shot for a lifetime or taken on a yearly basis like flu shot? So Dr. Ravi, do you want to reflect that about flu and how what is likely to be the vaccination immunity after COVID-19 vaccine, considering the immunity or immune approach?
Thank you for the question. The honest answer is we do not know. But if you look at literature as we have summarized, the three of us have summarized, as of now, it looks like the virus is able to induce both humoral and a cell-mediated immune response.
Thanks, Dr. Ravi.
Natural infection, what we are finding is antibodies are waning between 3 to 6 months. So if we believe that that is going to happen also following vaccination, which is actually a aggressive way of pumping the immune system compared to natural infection, I would believe that at this point of time, for our own comfort, we should accept that it may not last long. Having said that,
Doctor,
it'll be very nice if vaccine can induce long-term immunity. The answer to this question will be available only next year. Till then, it will be speculation. And Raches can add to this, but my own gut feeling is vaccines for respiratory viral infections, generally, it's not lifelong immunity. Flu is a classical example, but that's an extreme example because the virus mutates very rapidly. Over.
thanks, Dr. Ravi. So, Dr. Ella, you want to add something?
Sure, I think Professor Ravi has summed it up extraordinarily well. I think we need to be a little realistic here. We shouldn't be over ambitious. We should understand the realities and complexities that go with COVID-19. And we have the best example. We're comparing all our vaccines to natural infection, and antibody humoral responses are plummeting 3 to 6 months post-infection. So only time will tell, and science will dictate that answer. And at the I would just completely agree with Professor Ravi's answer on that.
Thanks. Thank you very much. So, thanks. I really appreciate your response, and also appreciate the fact that you both of you were really brief. There are many questions, and my co-panelists can see. I would request Dr. Ella to talk about that. Some of the specific questions which are about the vaccine work which he's leading, and these questions are related that is Bharat Biotech planning to apply for emergency use authorization after the phase 2 data analyze and results available. So can you please answer that question? But at the same time, I would request that we need not to be very specific in these responses, and if we can lay down the broader principle rather than answering specific for the particular vaccine. Thanks.
Sure, definitely. That's a great question. I mean, emergency use authorizations have been granted to a few, I would say, therapeutics such as remdesivir and hydroxychloroquine for a certain amount of time. With regards to vaccines, I don't think that's the right approach. I think it's very important to understand the immune response and what does the immune response translate to. And the best measure is to understand clinical outcomes rather than immunological outcomes. How much is your antibody response, your ELISA, and your neutralizing capability is all science jargon. It needs to translate to real world settings. And that's where a phase 3 trial will answer these questions. So I don't believe in that personally. And even Bharat Biotech doesn't believe in that either. That's why we're entering phase 3 clinical trials, which will only look at the efficacy.
Thanks, I just want to add to what Raches beautifully summarized. From a common sense perspective, the topmost priority for a vaccine should be safety. It should not harm anyone. Second, it should protect against disease. That is what we want, most important. The bonus is if it can prevent infection as well. Over.
Yeah, thanks. So we are hearing from the speakers, and I would like to add an additional point here that while the regulatory authorities in each of the country have their guidelines and they have procedures to give emergency use authorization for vaccines and other products. However, in times such as pandemic and other, it's key that we use these very rationally and these are not overused. So the countries have a provision, and some countries have done that process, but that should not become a routine and not without the due diligence and the continuous follow-up and monitoring the safety record of the vaccine as and when such processes are done. So if a provision is available in national regulatory authority approval, it does not mean it should be used, and that should be the approach. Moving to the next question, which probably Dr. Ella can answer, and this question is about what would be the packaging, whether it will be single dose vial or multi-dose vial, and also some reflection um, on the cold chain capacity and cold chain temperature requirement for the vaccine.
Sure, very valid, pragmatic question that is. This is an intramuscular vaccine, as most vaccines are. It's a two-dose regimen given with a 28-day interval. So if you get the first dose of the vaccine, you wait for 28 days, you get the second dose of the vaccine. With regards to packaging, these are usually ready-to-use liquid formulations, either in a single dose or a multi-dose vial. What we are looking at is definitely doing multi-dose in the need of a pandemic, and to reduce the burden on the cold chain and to make it easier. I mean, it's easier to send 5 vials in a glass vial rather than one vial, one dose in a glass vial. They occupy the same amount of space. It's more efficient, and I'm sure Dr. Lahariya can add to the benefits of multi-dose presentations.
Thanks, Dr. Ella. Well, I think, of course, if it's a government supply and large scale vaccination need to be done, then it had to be multi-dose vial. That saves on a lot on the cold chain space and also on the transport and other costs. So for pandemic response, more likely to be multi-dose vial, but how many doses per vial, that will be logistic issue and will be determined by many other factors. So I'll quickly move to the next question, which is for Dr. Ravi. So this question is that how long the virus stays in mouth and nostril before it enters and causes the disease?
I don't know if people have read the Joint Commission Chinese report, which was the first one out, and subsequently there have been many studies. The incubation period in 90 to 95% of the people is 5 to 7 days. But it can be as late as 14 days and very, very rarely beyond 14 days. So it takes on an average 5 to 7 days for symptoms to appear. That means if I got symptoms today, 31st October, I probably got infected on the 23rd, 24th, or the 25th of October. And that's the time the virus gains momentum by replicating in the nose and the throat, and eventually will go down to the lung. So that would be the answer. Over.
Thanks, Dr. Ravi. So this next question is about how long the antibody present in the beneficiary. Maybe I'll try that answer or that question. So the studies available till now has indicated that the person has a detectable level of antibodies anywhere ranging from 60 days to 90 days. However, whether antibodies, disappearance of antibody means no protection, that we don't know for sure, because what we are hearing and what we know that most of the vaccines which are being developed and also natural infection results in T and B cell immunity, which means the memory cells are developed, which are only counted few, but they when the next infection happens, they quickly multiply and they provide protection. So currently the antibody level declines, which happens with all other diseases, not only with this disease. So, but the person develop a T and B cell immunity. We don't know the protective level of antibody in this case. We know for sure, for some certainty that in absence of antibody also person could be protected for that reason. So that's all we know, and this our understanding is continuously evolving. What is also equally important, and we know that the antibody developed by natural infection and antibody developed by the vaccination are very different, because in the vaccination we follow the boosting approach also, that you give first dose which is the priming part, and then we give second dose which is a boosting of the antibody. So the antibody developed and immunity developed after vaccination is likely to be very different than the natural infection, and we will keep learning about this as we go along. So that's a summary answer. If any of my co-panelists want to answer or chip in? Okay, thank you. So then I'll move to the next question, which is on which is almost similar, that how long the vaccine will be effective. I mean, 6 months or 1-2 years. This has already been answered. For Dr. Ella, there is a question that, are you planning to publish the results of phase 1-2 trials? Will the detailed clinical trial protocol be made public?
Yes, of course. I mean, with any of our clinical studies, we always get them published. We have around 150 publications from our various forms of research. And I myself take a lot of effort in the medical affairs department. And we are, in fact, in the stages of sending this out for peer review publication as well, and will be in public domain. That's phase 1 to phase 3, which will include our protocols, as any publication is, requires that.
Okay, there is another follow-up question, and which is related. So this is specifically asked about the adverse event due to various COVID-19 vaccine. You don't have to you need not to be specific on one vaccine, but in general, what you are hearing, what are the common adverse effect events which are being reported with vaccines?
Sure, definitely. I can give you from a non-Bharat Biotech perspective as to what I'm seeing in the literature. Many of these vaccines are quite novel, and they're given at high doses. These are live viral vectored vaccines, inactivated vaccines, and mRNA vaccines. And usually what we're seeing is that the reactogenicity, that is the immediate side effects generated from these live viral vectored vaccines and these mRNA vaccines, is quite high as compared to the inactivated vaccines as well. Please note that I'm not giving anything regarding to Covaxin specific. That data is being generated. We're not involved with the data analysis of that, as we're the sponsor. But this is what I'm seeing in literature, is that the adverse events is a little bit higher for the novel vaccines. For the conventional vaccines, it's a little less. But please bear in mind that any vaccine that sees licensure they'll very well make sure that the vaccine is safe, period. So I don't think we should stress on that too much if it is licensed.
Thanks. So, Dr. Ravi, I have a question for you that since you talked about epidemiology and then you have worked on viruses, that what do you foresee for coming winters and how this SARS-CoV-2 likely to behave in the coming winters, especially in India, and any reflection on second wave in Europe, and what do you think likely to be course for India?
Okay, I can speak from my experience. First of all, India is not one country. Let's get it very clear. We are 30 countries and we are 30 different cultures, and introduction of SARS-CoV happened into different states and districts and rural areas at different times. So the short answer is, it is going to be an evolving picture in each state. I can talk about my state. We've just finished a large scale survey. For the first time, we have done both antibodies as well as RT-PCR and antigen, something which has not been done in other surveys. And I can tell you that, not to be quoted and not to be definitely put on WhatsApp or in the press, a third of the population seems to be already exposed to the virus. Okay?
Thank you.
So my specific answer to the question is, all respiratory viruses come in wave forms. The first one is huge. Majority of the people get exposed to the virus. So the virus can't find a niche in the population till susceptible population builds up in the community, and then there will be a second wave. What promotes the second wave? Definitely the most important is crowding and letting guard down. That is number one. Does weather have a role? For respiratory viruses, yes. The air in winter is heavy and it does not get cleared very fast. So generally, respiratory viruses, you will expect a spike in winters. Over.
Oh, thanks, Dr. Ravi. So there is another question, which is whether vaccine will be 100% preventive, and this is question which is often asked. People are looking for an ideal vaccine, and only ideal vaccine can be 100% preventive and 0% adverse event. But in the world, there is no ideal vaccine. Like some of the really well performing vaccines, measles vaccine provide nearly 95% protection after 2 doses. Hepatitis B vaccine is a really good and effective vaccine, but even with that kind of vaccine, the protection is 95%. What we need to understand that the efficacy is not entirely dependent upon vaccine, but also on the host factor. The individual may not develop sufficient antibody due to some immunocompromised issue and other aspect of individual specific behavior. So there is no such thing as a ideal vaccine or 100% preventive and 0% side effect vaccine. And that's where we need to keep our expectations realistic in the COVID-19 vaccines also. Because what we know that there would be vaccines, what we also know that these vaccines would be relatively less efficacious than all other existing vaccines, because these are the first generation vaccines, and first generation vaccines are developed to ensure maximum safety and then check the efficacy of those vaccines. Once the vaccines are available, then effort scientists and vaccine researchers and vaccine manufacturers work on various aspects to boost their efficacy through adjuvants and other approaches. So in this entire process, it will be key that we keep expectations real. And that's what is happening at global level, that most of the national regulatory authorities have said that a vaccine which is more than 50% efficacious would be licensed. And I guess that is a good starting point for a pandemic response. Even with the 50%, what we know that every one of the two people vaccinated will be protected, and which would definitely accelerate the process to have a population with the sufficient immunity and get us out of the pandemic. Another related point here is that there would be more than one vaccines against COVID-19, which we know. Some would have, and each of these vaccines are likely to have a different efficacy. And not only the different vaccine likely to have different efficacy, the same vaccine in different setting might have a different efficacy. So we need to be prepared for such eventuality, and without being comparing those vaccines, all those vaccines which meets a certain criteria of efficacy would provide the protection and need to be used, and that's what our purpose should be. Any thought from my co-panelists, Dr. Ella?
Just two philosophical points. My teacher used to say, to live is to risk dying. There is no 100% for anything in life, whether it's a diagnostic or a vaccine. Second point, I would reiterate what I showed in my slide. The greatest microbiologist in history, Louis Pasteur, said, the seed is nothing, the soil is everything. The vaccine actually may be extremely good. If my immune response is not appropriate, it's not going to protect me. Over.
Thank you. Well, so we are approaching toward the end of this webinar. What I'm going to do, I'll come back to my co-panelists for one last thoughts. But before that, as you can see some of the questions on the right-hand side of the chat box. So I invite you to look at those questions, and if you want to answer some of those questions which I have missed, because they have been asked in different approaches and different forms, or unless there are more new questions, then we will move to one final round of concluding remark from each of the panelists, and then I'll conclude toward the end. So please have a look at the sidebar chat box, and if there is any question you want to respond, else we will move to final concluding remarks.
Sure, there is one question. I think a doctor was asking us about our clinical development plan and why we had changed our sample size. While we were doing the phase 1 trial, we decreased the phase 2 sample size. I would just like to bring to the doctor's kind attention that we are conducting something which is called an adaptive clinical trial. So under one protocol, we are allowed to make changes as we go along the way, as we get new data, as we understand the safety profiles, the immune responses of the vaccine. We're able to make necessary changes along the way. That could be with sample sizes as well. And this is all done ensuring that our statistical power, which is 90%, is always adhered to. So that's the prime reason why we've shortened our sample size. And as you can see here, there's no cause for concern. We're doing a 26,000 subject size phase 3 clinical trial, and we're on par with the other Western manufacturers. Everyone seems to be doing an efficacy trial between 25,000 to 35,000, and we're somewhere there as well. So in that regards, everything is adaptive. It's called novel coronavirus for a reason. We're learning a lot of things as we go along this process. Thank you.
Ravi?
No, I don't have any questions that I want to give inputs on.
Okay, so that brings us to the final section of this webinar. So I invite you for 1 minute concluding remark or summary. So maybe we start with Dr. Ravi again.
Yeah, it is a novel virus we are learning. I'm very proud of the Indian vaccine industry. Extremely proud. I've always been proud. They've come forward very briskly and quickly. I'm sure we will have vaccine. Until then, use the social vaccine. Maintain distance. Definitely wear masks. The NEJM paper is out. It clearly shows that masks are effective. Wash hands. Be safe. Thank you.
Dr. Ella.
Sure, definitely. As Professor Ravi alluded to, Indian vaccine manufacturers are very strong. We supply majority of the vaccines to the rest of the world. We're talking about low and middle income countries. There are around 100 of them. We predominantly supply all of the vaccines. Around 125 million children are born each year. Indian vaccines touch at least 90 million of them. So that's the impact we have on a global scale. We can deliver for COVID-19. Hopefully, our results will be promising and we'll get this vaccine as soon as possible. But I think it's very important that we be patient. It's very hard to roll out 16 billion doses of vaccine. And here is behavioral changes. As you know, you're all physicians. You inculcate behavioral changes in your patients. But this has to be done as well. And as Professor Ravi mentioned, continue to wear the mask, continue to do social distancing. Even if the vaccine is out and if it's licensed and it's out in the news, please continue to wear your mask. And as medical professionals, please carry the word to your patients as well, just to keep your community safe. Thank you.
Thanks, Dr. Ella. I would like to echo whatever both my co-panelists had said. We are hearing, and right now we are in mid of the pandemic. What we know that pandemic are the reality, and being prepared for the pandemic is what humanity should do, and which we have all of us have done. A few things has emerged from this challenge which is posed by pandemic, which does provide us opportunity to convert this challenge into strength and opportunity. What we have learned that health has become a mainstream discourse across the country, from top level political leaders to person on the street is thinking about health, and this is an opportunity to strengthen Indian healthcare system. What the pandemic has also brought to the forefront that while we focus a lot upon medical care, and many of you in the audience and the participant in this webinar are physicians, that medical care is important, but what is saving the day from the pandemic is a public health intervention, the preventive and promotive intervention. So we are talking about much of our success when there is no therapy available, when there is no vaccine available, is dependent upon the non-pharmacological interventions of face mask or face cover, hand washing and hand sanitization, and also social or physical distancing. And these are the preventive and public health measures which can work to prevent many other illnesses. For example, sanitization can reduce the child mortality drastically. The social distancing or face mask can reduce the burden of tuberculosis. So these are the broader public health interventions. and preventive measures which are we should uh, carry forward and continue to adopt in the time ahead to make the society healthy. Uh, the vaccines which we want so much today, and an entire world want one vaccine, which is COVID-19 vaccine. But at the same time, we also know that even the available vaccines are not sufficiently used. In India, children do not receive the already available vaccines, recommended vaccines for them. The use of vaccines for adult population, including high risk population, is so low. So as we are thinking and talking about COVID-19 vaccine, it is also time to reflect and think that how to increase the utilization of existing and available vaccines for the target population. And fourth, it has brought the science to the forefront, and the scientific thinking should also be brought in our daily life and approaches. So there are many opportunities which can emerge from the challenge which we are facing, and we should carry those forward. Thinking health holistically from preventive, promotive, public health, as well as medical care. With all these words, I would like to thank my co-panelist, Dr. Ravi, Dr. Raches Ella, and also the Practo team for organizing this webinar. This was a really useful, and I found this conversation really helpful. I hope the participants who have attended this webinar would have enjoyed. The conversation has just started, and we need to continue this forward. Thank you very much. And with this, we can close the webinar. Thank you.